Hello, today we'll start studying different types of biomedical signals, their sources, and how they can be recorded or stored. So, in this lecture, we'll focus on electrocardiogram. So, now we'll see what are the different types of biomedical signals and their sources. So, there are different types of biomedical signal sources are there, like we can have biomagnetic signals which is generated from encephalogram, magnetoencephalogram. Similarly, we have some ocular system that can be recorded from using this electrooculogram. Similarly, heart is also generating some electrical, there is some electrical conduction path, and it is also giving some action potential that can be recorded through electrocardiogram, that is a bioelectric signal. Similarly, we have bioimpedance signal such as your impedance nemography is the way to record this bioimpedance signal. Similarly, biomuscular system or bioelectric signal generated by the muscles or muscular system, that is recorded through electromyogram EMG. Similarly, we have other signals which is generated by pulmonary system, which like your pulse oximetry is also another way to record this pulmonary optical signal generated by this pulmonary system. Similarly, we have another biooptical signal that is related to a cardiovascular system, which is your pulse rate. Similarly, the brain waves is also generated by the nervous system of the brain, which is electroencephalogram. It is also another bioelectric signal. Similarly, respiratory system, esophagus, temperature, these are the some different types of parameters, or they are acting as one different types of signals, giving the activity, giving significant activity and their functionality of proper functionality or normal functionality of our different types of physiological activity at different organs. Similarly, heart sound, which is coming under bioacoustic signal, it can be recorded through this phonocardiogram. Then we have the major time we are measure this biomechanical system or biomechanical signal, that is your blood pressure. It is related to your or generated by this cardiovascular system. Similarly, another cardiovascular system, that is your biomechanical signal, that is blood flow. And also bioimpedance, it can be taken from the skin impedance, it is a galvanic skin resistance actually. So, these are the different sources of biomedical signals and how to capture them, like your electromyogram, pulse oximetry, then we have electroencephalogram, electrocardiogram, electrooculogram. All these are different types of biomedical sources and the recording systems. Now, as we have discussed, these bioelectric signals, they can be generated from different nerve cells. The major part of this bioelectric signals are generated by the nerve cells. And muscle cells. So, the examples are electrocardiogram, electroencephalogram. Similarly, bioacoustic signals, acoustic means sound, it is related to sound. So, the flow of blood in the heart and the heart valves, they are actually during this pumping action of the blood inside the heart. So, the valves, the closing and opening sound is generated by this valve closing and opening. So, this creates a lobbed up sound that is coming under this bioacoustic signal. Similarly, we have biomechanical signals which are related to motion and displacement, pressure, flow signals. Biochemical signals are the partial pressures of gases like carbon dioxide, oxygen, and concentration of various ions in blood. So, these are all biochemical signals. We have biomagnetic signals. In biomagnetic signals, there is a very negligible amount, means extremely weak magnetic field are produced in various organs like brain, heart and lungs. So, those are called magnetoencephalogram signal, which is generated by the brain. Similarly, we have biooptical signals. Biooptical signals means we are actually passing some optical energy to capture the different which will scatter through the at different tissues, and we capture those refracted and the scattered signals, and we from that we can measure some or significant change with respect to the reference. So, these are called biooptical signals. Similarly, bioimpedance signals. Impedance, you know that the impedance it is like it is providing some resistance when the current is passing. So, impedance of the tissue, it is different depending upon the composition, blood distribution, blood volume at different parts of our body. So, that can be one signal. And this measurement of the galvanic skin resistance is typical example of this type of signal. So, next we move to the heart electrical signal conduction, and we will discuss how this electrical signal conduction or generating the action potential. This action potential is important for the proper conduction or proper pumping action of our heart. This is started from this SA node. We will see one by one. This is a SA node, or we call that sinoatrial node. This SA node we call that pacemaker, natural pacemaker. It generates one action potential. That action potential, we know that previous classes we have already studied what is action potential. Action potential is created or it is generated due to the depolarization of the cells, because previously at this place we have a resting potential. Then once this SA node is generating the signal, it will depolarize the cell near it. Then that electrical action potential or that potential will travel all over this heart muscle. So, mark it will, this heart muscle contracts in response to the electrical stimulus which has been received and generated electrical pulses and conduct them throughout the muscle heart and stimulating the heart to contract and pump blood. So, all this started from this SA node. Then it will, you can mark from this, the signal will travel to this path, this interior internodal path, that will send or through those internodal path the signal which has been generated or the action potential which has been generated at the SA node, that will travel to the atrioventricular node. This signal will again, that will travel or pass through this middle internodal path and then moving to this ventricular part, and all over the ventricle you mark, it is covered with this nervous system or this branches. We call that at this part it is called bundle epiphyse, and it will going to spread all over the myocardium, which comprises the heart muscles, and all over that myocardium will find this signal will travel through this Purkinje fiber. So, this signal transmission, this electrical signal transmission which has been generated at this sinoatrial node, it will take some time to travel through this path. So, you can mark here it if this is your SA node, from here it starts, it will take some time to reach at this at least 0.12 second, it will reach at this AV node, then it will travel through this branches in both myocardium part or both left ventricle and right ventricle, and they starts pumping. So, these are the main electrical potential or the action potential, that is depolarizing the atria, then that depolarization continue to depolarize the ventricles, then once this ventricle, both the ventricles are depolarized, then again the repolarization process starts and this ventricle and the atrium, they become repolarized. So, this is the process through which the pumping action of our heart is going on. So, now this particular rhythm, this action potential which has been conduction, which has a conduction path as we have discussed, that creates one rhythm of this electrical pulses or waveform. So, we call that sinus rhythm. Okay. So, this will start from the sinoatrial node, that is your natural pacemaker. Then this SA node, this SA node is present at the upper part of the right atrium wall. So, in the right atrium your SA node is present at the upper part. Then it generates electrical impulses and which initiates the each heartbeat. Okay. It will generate it as a rhythm. Signal pathway, as we have discussed, it will travel, this signal will travel to the atrioventricular node. Then this atrioventricular node, it is located between the atria, means the lower part of the atria and the top of this ventricle, means starting of the ventricle. It acts as a gatekeeper and delays signal so that atria fully contract before the ventricle. So, after this AV node, signal passes to the ventricle via special conducting fibers, as we have discussed, the Purkinje fibers in the lower heart walls. Then this signal transmission generation, it is controlled by this autonomic nervous system, which regulates sinus node firing and can double heart rate within 3 to 5 seconds during exercise, meet increased oxygen demand. So, it is not under our control, it is completely under autonomic nervous system, under the control of autonomic nervous system. Now, this action potential which has been generated or triggered at this SA node will start what they are actually doing. Due to this action potential, how the different parts of the heart muscle or heart is depolarized, repolarized, all these things will happen, we will see step by step. This process is called your electrocardiography, the recording of this curve or recording of this electrical pulses or waveform, we call that electrocardiography. First, this wave, they have different peaks and different valleys. So, you can mark here in this graph, it is clearly written, we have it is we can visualize, we can clearly see that there is one peak here, it is your P wave. This P wave, this peak is called P wave and it represents the atrial depolarization. That means, when at the excitation started at this SA node, first it will depolarize the atria, because as we have discussed, the SA node is present in the top of this atria, right atrium. So, it depolarizes the atria. It occurs due to the sodium influx, as we have previously discussed in our previous lectures. In the action potential, we know that during the process of this depolarization, there must be some influx of sodium ion into the cell. So, here also sodium influx causes membrane potential positive and it will lead to some positive voltage. This leads to atrial contraction. We call the atrial contraction is known as systole. So, this action potential which is generated due to the sodium ion exchange or sodium influx into the cell or cells of this atrium of heart, they leads to the contraction of this atria. So, this process is called atrial systole, and this duration you can mark here. This duration will take, this is the duration, time duration, it will take it sustain for 0.11 second and the peak amplitude will reach up to 3 millimeter in height. So, this is all about your atrial depolarization. Okay. Next we can see here you have then this wave will continue and we reach at this valley which is known as the starting point of this is Q. So, this PQ interval, as we have discussed, it is your 0 point that we will discuss. Here it is from this valley, again it will rise to R waves and coming down to S. So, we are getting one peak, sharp peak of QRS. How this QRS will relate to our heart contraction. So, this PQ or PR interval, it is your atria to ventricular conduction. So, after that this region you will find here in this curve you will find PR interval is this much. It is from 0.12 to 20 seconds, because as we have discussed this region is 0.11 second in the previous slide we have seen that. Now, from this particular point to this point it will take 0.12 to 0.2 second and this is the starting of ventricular contraction or depolarization. This electrical signal travels to AV node, bundle of his and Purkinje fiber. This will slow down, this AV node will slow down or create the delay of passing of this signal to the ventricle. So, slow conduction through the AV node allows complete atrial contraction. Otherwise, without complete contraction, if the ventricular depolarization occurs, then complete blood volume will not come into this ventricle and it starts pumping it out. So, this will take the AV node allows the complete atrial contraction by introducing the delay before ventricular depolarization. But a prolonged this PR interval may indicate a heart blockage. If this length exceeds this range from 0.1 to 2.2 seconds, then there may be a chance of heart blockage. So, then only once it occurs and at this phase it reaches at this Q point, then ventricle fills with blood from the atria. Now, next part is next curve is your QRS complex. It is your ventricular depolarization. Now, you mark once the ventricle is filled with blood, another pumping action or depolarization, that signal which has been at the AV node, now it has been passed through bundle of his and Purkinje fiber to the myocardium of these ventricles. Now, the ventricle starts depolarizing, both the ventricles start depolarizing. Once it has been depolarized, we are getting one peak. This peak is your R, or this duration or this complex, we call it as QRS complex, that indicates the ventricular depolarization. Massive sodium influx produces sharp spike in the potential, because we know that ventricular depolarization requires a higher potential, because it has to pump blood to all over the throughout the body. So that is why it requires higher potential. So a massive sodium ion influx occurs, and due to that we are getting a high sharp or high peak, that is your R, in this QRS complex. It results in ventricular contraction. We call that ventricular systole. Previously we have seen is this region, it is your atrial systole. Now this R indicating this ventricular systole. Then atrial repolarization occurs simultaneously, but marked by QRS complex. When this ventricular depolarization is started or it is going on, simultaneously this atrial repolarization also started, because the signal has been transferred or it has been passed from this SA node to the AV node, and as it is passing away, means it is moving away from this atrium, then repolarization started, and this interval is equals to 0.05 to 0.1 second. The next is the ST segment, that is your plateau phase. Ventricle remains depolarized. In this phase, S to T you mark in this curve from S to T, there will be no much higher peak in this case, in this region. We call that plateau phase. Here ventricle remains depolarized because the pumping action is still on process. So influx of calcium ion, it balances the efflux of potassium. Influx of calcium balances the efflux of potassium, producing a plateau. This process is very important. Sustains ventricular contraction to pump blood into the aorta and pulmonary artery. So as long as the complete blood which is present in the ventricle, they are not formed into this into the all of the body and the lungs through this aorta and pulmonary artery, till then this depolarized state will remain. Then comes the T waves. So now you mark in this curve. Then we have a smaller peak, that is your T wave. This T wave is giving you a ventricular repolarization, means the whole blood which is present in ventricle, that has been pumped away, and now it started a depolarization. It represents restoring the resting potential of the ventricular cells, and a potassium efflux returns membrane potential to a negative state. Then ventricles relax, marking the onset of diastole. So this process is called diastole. The T waves indicates or map to a diastole. Then TP interphase, electrical resting phase. Both atria and ventricles are electrically silent, we call it resting phase. Resting membrane potential maintained by the sodium and potassium pump using this ATP, and the heart refills with blood before the next cycle. So this process is your relaxation stage or diastole stage. Now this rhythm, this P, Q, R, S, then T, this rhythm repeats, and this is called sinus rhythm. This name is given normal rhythm of the heart where electrical stimuli are initiated in the SA node and are then conducted through the AV node, then bundle of His and bundle of branches, then through the Purkinje fiber. Now, in this table you will find one summary of the above discussion. Now, coming to the recording system or lead system of this ECG or of this action potential generated in this, as we have discussed in the previous slide. Now, this action potential, this waveform, that is PQRST, so it can be recorded through this electrocardiograph leads or electrodes, we can say. So, here they have this ECG, we call it electrocardiograph, it is your ECG, we call in short form ECG or ECO or EKG. So, they have 12 leads. Actually, it is based on a triangle, we call it Enthoven's triangle. It is the functional concept of electrocardiography, which has been developed by Willem Enthoven, who is a Dutch physician. This ECO or this ECG to diagnose a wide range of heart conditions such as arrhythmia, heart attack and other abnormalities in the heart's rhythm and functions using this Enthoven triangle. So, this Enthoven triangle it consists of 12 lead system. Out of these 12 leads, we have 3 are bipolar standard limb leads, 3 are augmented limb leads and 6 are precordial leads or chest leads. So, they are placed on the chest. Okay. So, now you mark here. The lead system are as follows. Here the first we have this 3 bipolar leads. Bipolar means we have taken a positive and negative lead and we are taking the difference of them. That difference will be recorded. The difference among them will be recorded. So, here you have, this is your right arm, left leg, and this right leg is considered as the reference point. So, this right arm, left arm and left leg. Here you will find, you can mark this arrow. This is your lead number 1. Similarly, this is your lead number 2 between this right arm and left leg, and this is your lead number 3, that is your left arm and left leg. So, these 3 are known as your bipolar limb leads. So, we will see more on the next slide, and these 3 augmented limb leads, they are just placed in this manner. You can see here, this is your augmented vector F, augmented vector L, augmented vector R. So, these 3 are placed in this manner, and what is this augmented limb leads? We have slides in the later page. So, in this case you have, other one is your precordial leads, which will be placed. There are 6 precordial unipolar leads. These are unipolar leads. They are placed on the chest as it is shown here. Now, one by one we will see. Here these 3 leads, lead 1, lead 2 and lead 3. Lead 1, lead 2 and lead 3, they are actually your limb leads, okay, or bipolar limb leads. And as we have discussed, the difference has been recorded between this left arm, right arm, then lead 2 is between this left leg, right arm, then your lead 3 is between left leg and right arm, and they are placed in along the shoulder 0. These 2 are placed 0 degree, they are at an angle of 60 degree in the lead 2, and lead 3 they the angular displacement is 120 degree. They are looking like they are just like a triangle. We call that Enthoven triangle. It is an imaginary formation of three limb leads in a triangle used in electrocardiography, which has been formed by the two shoulders and the pubis. The shape forms an inverted equilateral triangle like this, as we have seen here. Right arm, left arm and left leg. Okay. With the heart at the center, that produces zero cardiac action potential, means this part is the center and this is acting as zero potential, or it is your reference, you can say, when the voltage are summed up. The vectorial addition will give you a zero potential at this point. So, it is acting as one reference point during measurement. Now, next one is your unipolar leads. Among these unipolar leads, we know that one is your augmented leads. So, this when this electrode are placed on the chest near the heart, higher potentials are detected, rather we are placing them in the limbs, which is away from our heart. So, this led Wilson in 1984, he introduced a unipolar leads, where the ECG is recorded between a single exploratory electrode and a central terminal representing the body's average potential, and formed by combining several electrodes at one point. They are called augmented leads or averaging leads. Now, we have augmented vector right. This is your augmented vector right. Here you have taken these two and this is the average between them. That means right wrist versus the average of left arm and left leg. Similarly, in the lead of augmented vector left, the difference we have taken, the potential we have taken measure, that is between this left wrist versus the average of right arm and left leg. Similarly, augmented vector foot, this is your left foot and versus this is your versus right arm and left arm. So, through this they can measure different potentials. And along with this, we have those precaudal potential or that limbs. So, here you mark, we have the waveforms for three different leads, lead 1, lead 2 and lead 3. So, actually if you mark carefully, this lead 1 and lead 3 will, if you add them vectorially, they will give you this waveform, your ECG or electrocardiogram. Now, here we have, this is in the lead 1 we have taken this AVR, lead 2 AVL and lead 3 we have considered this AVF, and the chest or the precaudal limbs electrodes, they are in the limb 1, we can find it V1 V4. Similarly, it is your V2 and V5 in the lead 2 and V3 and V6 in the lead 3. So, this is your complete ECG curve representing the PQ RST curve. Now, from this curve, why it is important? Because if the particular rhythm is not followed, that is 72 beats per minute, if it is not followed, then we call it as some abnormalities of heart. Now, this is your sinus bradycardia. It is the slower heart rate. If the heart rate is below 60 beats per minute, then we call that sinus bradycardia. You can see here, this is the curve. Similarly, sinus tachycardia, it is a higher beat than the normal. So if the beats that heart rate is more than 100 beats per minute, then we call it as tachycardia. And next one is the sinus arrhythmia. Arrhythmia means the repetition is not uniform. So here you mark the repetition, here we have 1, but the repetition is twice, then there is no pulse here, then again pulse. So these are this arrhythmia. So this type of arrhythmia, one of the arrhythmia we can call it is atrial flutter. They will just waveform is like this. When this occurs, when the flutter contraction rate can be 200 to 300 per minute, so high. Another types of abnormality we will find, we can detect from this ECG, that is your atrial fibrillation. Atrial fibrillation means we know that when this absence of sinoatrial node stimulus leading to loss of P waves, if there is no strong SA means signal, that action potential at the SA node, then we cannot get this P wave on the ECG curve. This atria fibrillate or quiver instead of contracting effectively. So you can mark this wave, it will not contract, this muscle will just quiver, and the atrial kick is lost, decreasing the ventricular filling and reducing systemic and coronary blood flow by approximately 30%. So as this atria is not getting enough, or it is not depolarized properly, it will not contract and the blood will not flow, all the blood will not flow into this ventricle. The fibrillation or quivering that takes place due to the individual muscle fiber contracting independently. There is no sync or there is no coordination in between the muscle contraction on this stage. So this will due to the chaotic conduction within the atria. As you can see here, there is no proper path of this signal conduction, rather than they are chaotic in nature. So this atrial fibrillation has no P waves, as SA node is not functioning properly. Instead of a P wave, what you see in a number of small waves of different size known as fibrillatory or F waves. So instead of a P waves, we have F waves in the case of atrial fibrillation. So a defibrillator can be used. You can see here, this is the defibrillator. External supply is given to again coordinate or to again it has been applied to the chest wall, so that it can be again in a sync, or they will be synchronized. Now this is the block diagram of ECG machine. As we know that we have different leads, right arm, left arm, right leg, left leg. All these leads are they are connected to a protection circuit, means isolation circuit. This isolation circuit, it will not allow the direct current flow because these leads are connected to our chest. So there may be some shock hazards. So we have to use some isolation circuit, that is a protection circuit. Then through this protection circuit, they are connected to lead selector, which lead will collect, which lead is activated at what time, so that we can take the output or record the potential. Then this lead system, after lead system we have calibration circuit and fault detector circuit. Once the calibration has been done, that signal has to be put into an amplifier. This instrumentation amplifier is also known as pre-amplifier. It will pre-amplify this feeble signals and it will amplify that. Then once it has been amplified, we have to filter out the noises or motion artifacts Those, after filtering out this, then we again amplify that signal. Then we can use this amplified signal, we can take that for processing, we can use some microprocessor or computer, or before taking that to the microprocessor, we can connect here some analog to digital converter. Then that digital bits will be sent for processing to the microcontroller, or we can directly send that to some chart recorder or oscilloscope to record that. So, it has a 3 stages of this ECG machine. First is one input stage, then second was a signal processing stage, third one is output stage. So, this is the manner we can record this ECG. Okay, thank you.